Sorry.
So can we assume a good percentage of students have joined? And where we left it yesterday is the causes of cancer and the expression of antigens for which the immune system is triggered to respond. And then we covered briefly on the approaches of immunotherapeutic models, which have now reached the clinical level. And we saw examples for each case where the monoclonal antibodies have been used with a lot of success in the treatment of cancers. One thing did I not tell you yesterday, which we need to stress on today, perhaps because I made an assumption previously, you could have also covered part of the tumor immunology, is a number of oncogenic agents trigger development of cancer. Yesterday, we did mention only a couple of viruses which are associated with cancer, but you have to note that exposure to a lot of carcinogens do trigger the mutation in the genes in cells in the body, and then cells are made to proliferate uncontrollably, which we defined yesterday to be tumor further classified either to be malignant tumors or benign tumors. I don't need to go into detail in that because you learn much more about them in anatomic pathology. So again, in anatomic pathology, you could have gone through other triggers of cancer. I think also in hematology, apart from genetic mutation, which are associated with the inheritance patterns of genes, there are a number of carcinogens which can expose somebody to genetic mutation in a particular cell, which now triggers the development of cancer. Typical example, we have got chemical carcinogens. We have got even exposure to asbestos. We have exposure to ionizing radiation. So all these, I think you have covered them when you are dealing with the tumor immunology in your second year. Now, from today, we need now to narrow down something, which I need now your head to be a bit refreshed. We are looking at now the techniques which are utilized for the detection of those cancer antigens. And we understood yesterday cancer antigens can be tumor products or enzymes which are secreted when a cell is proliferating uncontrollably, and therefore define what we talk about as cancer. Yesterday also, I must have mentioned to you that, no, you can't see the slide, just be patient. You can't see it. Just be patient, I'll sort out that. So we also talked about yesterday about the cancer antigens which can be found in circulation. So these you could easily do serological tests and detect them. Today, I'm not going to cover that aspect of the serological testing because then it is the same principle which you are learning about ELISA detection of antigens or detection of antibody. In case of infectious agents, you are detecting the antibodies. In case of the antigens, like today, we are looking at the tumor antigens. If you have to detect these tumor antigens in serology or serological technique, you'll be using ELISA. So it cuts across the board the same principle which ELISA is used to detect the pathogens, like an antigen, not detecting the antibodies which are produced. So with that, therefore, I believe there's no reason for us to dwell on covering the aspect of ELISA in detection of cancer antigens, like I made it understood yesterday to you, the CA15, CA19, or PSA. You can do serological analysis and then detect their elevated levels. Here today, we are going to focus much more of now the solid tumors, and we look at how they can be diagnosed or detected. But later, I'm going to take you also through the technique of flow cytometry, which is used for immunophenotyping of leukemias. So even though we are going to dwell much more on the solid tumors, we will cover the aspect of the flow cytometry, which is also used for the immunophenotyping of tumors. Now, let me try, because if you're not, are you able to see the slide now? Hello?
Yes, we can see the slides.
Okay, so the first technique which we want to dwell on is the technique of the immunohistochemistry. And immunohistochemistry, perhaps I'm not going to go into refined definition of it. We are just talking about here immunology, histology, and chemistry. Then a student will be wondering, how does immunology, histology, and chemistry combine? We are looking at the antibody-antigen reaction, then immunology, histology here. So this antigen-antibody reaction in tissues come histology. Then the chemistry aspect of it is the kinetics of this antigen-antibody reaction. Combined, then you'll be talking about immunohistochemistry. In analogously, you can also talk about immunocytochemistry if you are intended to detect these cancer antigens from the cytological specimen, maybe fine needle aspiration cytology, pleural effusion if you aspirate. So in that case, you'll be talking about immunocytochemistry instead of immunohistochemistry. So here comes a bit clear. So there are a couple of techniques which could be applied to detect those antigens in tumors. One, which we are going to cover today, the immunohistochemistry. Then a couple of techniques also align in the detection of these antigens, which include immunofluorescence. Immunofluorescence is the same as immunohistochemistry, except the antibody which will be used to detect this antigen is fluorescently labeled with a fluorophore or a dye. So you can just master the concept of immunohistochemistry, but note that in the immunofluorescence, you will be using antibody which is conjugated to a fluorophore, which can now be interpreted in the fluorescent microscope. For immunohistochemistry, you just use light microscope to interpret the results. Then later, I think on 18th, if we have the lecture, we'll be covering the PCR detection of the both the genes which are mutated, as well as detection of, say, the pathogens themselves. You can be detecting, say, Epstein-Barr virus using PCR, as well as detection of the genes which are mutated, which now can confer advantage to the proliferation of the tumor cell. Western blot does not have much applicability right now in the detection of tumors, but you'd be thinking like comparing the proteins which are secreted in tumor cells and seeing the quantity of the kilodaltons of this protein. Now, the amount of kilodalton can tell you if there is a mutation in that gene, which can result into overt production of the protein, or if there is no production of that protein. Then I'll also be taking you through another technique, in situ hybridization. And in situ hybridization, while we use monoclonal antibodies to detect these tumor antigens in immunohistochemistry, for the case of in situ hybridization, we use probes. Probes are short DNA or RNA sequences which are utilized not in the PCR amplification, but now you are detecting that gene which is mutated in situ. I'll show you, I think, a slide, but I've retained that slide. I think I'll show you on 18th when we'll be going deep in detail of the PCR and in situ hybridization. Then I mentioned to you also about flow-activated cell sorting, which can be used for immunophenotyping of leukemias. But again, you can also use cytogenetics when you are analyzing hematological tumors and checking the genes which are aberrantly mutated and confer advantage for the proliferation of that cell. I know your minds are still growing, but you need to learn also about some element of gene sequence analysis. Yesterday, I told you that a tumor is a clonal proliferation of a single cell. So when you do sequence analysis or the clonality assay, you are typically checking the peak of the bands which are demonstrated when you are doing this gene sequencing analysis. In that aspect, therefore, two things can you learn. One, you can do gene sequences and mark the portion of the gene which is mutated. But again, you can also, under gene sequencing, do what is called clonality assay. And clonality assay is going to help you to see if what you are dealing with is a reactive process or or a malignancy process. So if you have got a reactive process, then the bands which are going to be manifested is completely different from what you are going to observe when there is a malignancy process. But the basis of immunohistochemistry is analyzing the structure of antibodies. On antibodies, you have got the constant and variable fragments regions, the crystallized fragment, which is used to classify the antibodies into immunoglobulin A, immunoglobulin E, immunoglobulin G, and etc., etc. Then you have got the antibody binding portion, which is upper. Where's the marker? I don't know where the pencil is. So you have got the antigen binding portion, and that antigen binding portion is the one which is critical for binding to the antigen when you want to locate the tumor antigens in the tissues. And in immunohistochemistry, you'll have got two techniques which are applicable right now. I'm not going to go into detail so much because you are not a technical staff. But the first technique which is applicable is called the direct technique. In this case, an enzyme labeled primary antibody will react with the antigen in the tissue. And then you have got the this primary antibody conjugated to a chromogen or a substrate. And when you challenge that substrate with an enzyme, peroxidase most of the time, then you find now the reaction between the substrate and the enzyme forming a product which is now easy to interpret when you have the slide under the light microscope. Challenge with this direct technique is that it has got low sensitivity and is usually used only with immunofluorescence. I've explained to you that immunofluorescence is just typical immunohistochemistry, but you are using fluorescently labeled monoclonal antibodies. And this is what we talk about the direct method. Here you have got an antigen expressed on the surface of the cell, and you're using the primary antibody to bind to that antigen. So I've mentioned to you that this primary antibody is labeled with a chromogen. Actually, it's not labeled with a chromogen, labeled with a peroxidase, which is an enzyme. So when you bring the substrate, then there is a reaction between the peroxidase and the chromogen reaction, and will be visible under the microscope as a complex. Then you have got a two-step, which is also termed indirect method. And here, an unconjugated primary antibody. While here, the primary antibody is already labeled with the peroxidase, in the indirect method, the primary antibody is not labeled with the peroxidase, but it is the secondary antibody which will be labeled with the peroxidase. However, the crystallized fragment portion of the antibody, which we've talked about here. We talked about crystallized fragment of the antibody H here. It has got a portion which will bind now to the secondary antibody to form a link. So it is the secondary antibody which will be labeled now with the peroxidase. And if you challenge that peroxidase with the chromogen or a substrate, there's going to be a reaction between that peroxidase and the chromogen to form a complex. So that complex is what is going to be interpreted when you observe under the light microscope. Typical example I'll give you when we go ahead. So when basically a primary antibody would be raised in a rabbit or a mouse, then the secondary antibody must be directed against rabbit. So we talk about monoclonal antibody against that rabbit. So secondary antibody against rabbit, which was the primary antibody. And yesterday, I must have explained to you, I'm not sure if I'd sent you the notes, how were these antigens detected, tumor antigens? You could have an experimental tumor antigen, then inject it in a rabbit or a mouse model. Then with time, you find that rabbit or mouse model developing tumor. So then you can utilize antibodies which are circulating in that rabbit to detect that antigen. So it is typical thing which we are now looking today practically, that we have got primary antibody raised in a rabbit because you challenge this rabbit with an antigen. Then when now you get secondary antibody must now be directed against that primary antibody. So this is what we call the two-step indirect method, which is illustrated here. As opposed to the primary antibody, which was labeled the peroxidase, here, the primary antibody which is binding to the antigen is not labeled with the peroxidase, but the secondary antibody is the one which is labeled with the peroxidase. So the secondary antibody has got, utilizing its antigen binding site, binds to the crystallized fragment portion of the primary antibody. And at the end, you have got this peroxidase reacting with a chromogen to form now a complex which can now be interpreted under the light microscope. I'll give you examples of this, so you don't need to be worried. I'm just taking you to the principle and the concept of that. So typically, a variety of primary antibodies from the same species can be used with the same conjugated secondary antibody, and this does not need to bother you much because you're not in the experimental laboratory. And what is being done here in the two indirect step method compared to the direct method is just to enhance sensitivity of detection of that antigen which was expressed on the tissue. Sometimes you can also have a three-step indirect method, which is not quite much applicable nowadays in the routine practice. And here you could have a second tier. So you have that primary antibody not labeled, secondary antibody not labeled, but now you have got the tertiary antibody, which is the one which is labeled. In either case, what is important, which I've explained to you, all these indirect methods, you are attempting them so that you increase sensitivity of the detection of that antigen in the tissue. So this is what is explained. So sometimes you have got the antigen down here. The primary antibody has got its portion, which is binding to the antigen. And occasionally you can also find a double labeling of this peroxidase to both the secondary and the tertiary antibody. So this procedure is a bit more complex, and not only complex, it's becoming more expensive, considering that all these antibodies, if you have to generate them commercially, then it will be adding to the cost to the patient in the detection of that antigen. No wonder now what is only applicable is the two-step indirect method, because its sensitivity is equal to what you would have gotten in the three-step indirect method. So here I've explained, I don't need to go into why you have to label the primary antibody with the peroxidase, or why you must label the secondary antibody with the peroxidase, or if you got the three-step indirect method, why you have to label with the soluble antigen to form an immune complex, simply to enhance the detection or reaction with the chromogen or the substrate, which would subsequently form a complex enzyme substrate reaction, which will form a complex, and that complex is what you will now be able to interpret under the microscope. So sometimes you have got conjugation which runs, like I mentioned to you. This also a technique which has not, is not utilized currently because of its complexity. And overall, I mentioned to you, imagine you want to produce the primary antibody, produce secondary antibody, produce tertiary antibody, and even tertiary antibody, you want to conjugate it further to a peroxidase antiperoxidase complex. It becomes overall now expensive, which cannot be applicable in routine diagnostic laboratories. So all this I put to you when you make your personal reading. If you come across them, you note that these are techniques which were being used and applicable previously, but now not routinely used in diagnostic laboratories. However, you have to know the substrate and the enzyme. We have talked about the peroxidase as the substrate which is being used, but you have to know I've talked about peroxidase, the enzyme which is being used, but you have to note a little bit of the conjugation using the chromogen, which chromogen enhances these technologies are much better. So when you come across streptavidin biotin complex analysis, it's just typical glycoprotein, which is conjugated, like we are looking about conjugation here, antiperoxidase complex here. So you use conjugation with streptavidin, which is generated from Streptomyces avidini, and avidin, which is also extracted from the chicken egg. And these are only helping to enhance the binding to the primary antibody, which had already bound to the antigen to enhance its detection. And it looks now a bit more complex too. We have got biotin conjugated rabbit anti-mouse immunoglobulin, and we have got peroxidase enzymes, which I've shown you. So later, when you bring now the substrate, it gives much more enhanced detection. But this I've mentioned to you from the indirect 3-step or quaternary step in the detection of the immunohistochemistry. They are not routinely utilized. When you will be either rotating in histology laboratory or immunology laboratory, you will find the detection kit marked like ABC complex kit, avidin biotin complex kit, and it may be your worry, what does this avidin biotin complex kit mean for the detection of the antigen? So this is what I'm explaining to you here. Practical illustration, I think you'll be going through when you will be demonstrated to by the technical staff. And still in the complex imaging of the racing antibodies against each antibody species, making it antibody horseradish peroxidase or horseradish peroxidase. These antibodies, primary antibodies, can be interlinked with the horseradish peroxidase to ensure much more detection of the antigen. Look at where the antigen is on the surface. This much more complexity is only typically applicable to enhance the detection of this antigen, because a more reaction will occur when you have got horseradish peroxidase reacting with the primary, with the chromogen of the substrate. All that said, put it now aside, the principle of immunohistochemistry. So where do we apply this immunohistochemistry? One typical example of application in immunohistochemistry is immunophenotyping of malignant tumors, particularly the non-Hodgkin's lymphoma. So you may be having, like today, perhaps Dr. Mushiri was telling you about the morphology, how it could look like the starry sky pattern when you are encountered bucket lymphoma, suspicion of bucket lymphoma from the clinical presentation, then maybe FNA cytology is done and it's still not clear to you, so a biopsy is recommended, and you pick the biopsy, and you now want to detect the antigens which are expressed in these tumors which are characterizing the bucket lymphoma. So one typical example of application of immunohistochemistry is the immunophenotyping of malignancies, and typically the non-Hodgkin's lymphomas. We'll go ahead and give you example of its application in carcinomas or undifferentiated tumors, but this you can also make on your personal readings. So when you look, for instance, you get the morphology of bucket lymphoma having a typical lymphoid cells, a phased lymphoid architecture, prominent starry sky pattern with tingeable body, histiocytes and macrophages. So the cells would be looking like, oh, these cells may, under the microscope, be looking to me to be a diagnosis of bucket lymphoma. Now, you have to move ahead as recommended by the WHO to combine both the clinical presentation, morphology, immunohistochemistry, and molecular analysis to make the correct diagnosis of bucket lymphoma. So having looked at those round cells, nuclear, clumped chromatin, and all that, you now move on to be looking at describing clearly the morphology of that bucket lymphoma or bucket leukemia. Presentation of this tumor, I don't want to go into right now because you must have covered a little of it in hematology or in the clinics as you move. So here we come now to starting your immunohistochemistry and looking at the markers which are expressed during the development of these non-Hodgkin's lymphomas, and you narrow down to those markers which will be expressed predominantly by the bucket lymphoma or leukemia cell. The cluster differentiation 10, which characterizes germinal center tumor, and CD19, which characterizes mature B cell, which is almost exiting the germinal cell, and you have got the cluster differentiation 20. Other markers you can include CD45, CD38, CD5, or terminal deoxynucleotide transferase. So you would note that terminal deoxynucleotide transferase would be negative because bucket lymphoma is a mature B cell lymphoma. One marker which is
Dr. Nyangol? Dr. Nyangol? He's joining back.
Are you still seeing my slide?
No, doctor. Please, if you can begin the
No.
presentation again.
Because the power went off, so I had to switch to
Hotspot.
now.
Yes, we are back.
So we were here, and I was explaining to you there are two critical markers for the diagnosis of bucket lymphoma, one which is the BCL2 and BCL6. So I'll take you also into comparison when you are doing the bucket lymphoma diagnosis from the diffuse large B-cell lymphoma, because this you can learn with time. Don't cram these kind of things. This you learn when you'll be maturing in pathology, those who would be wanting to narrow in pathology, to know critically which markers can you request for analysis when you are suspecting a case of bucket lymphoma, when you're suspecting a case of, say, diffuse large B-cell lymphoma, etc., etc. So for the case of the bucket lymphoma, KI67 I've mentioned to you is almost 100% positive. The CD79, which is an early marker of B-cell, is also positive. You have also other markers, the PAX5, but notably, you have got about 20% of the bucket lymphoma cells staining BCL2 positive. But in ordinary cases, you will have this BCL2 negative. So this, if you find the BCL2 positive, please shift your mind towards now diffuse large B-cell lymphoma instead of bucket lymphoma. If you have KI67, maybe 40 to 50% positive, shift your mind towards the diffuse large B-cell lymphoma instead of bucket lymphoma. So overall, we are talking about the utility of immunohistochemistry in immunophenotyping of lymphomas. You can also use immunohistochemistry or analogously immunocytochemistry when you want to analyze the ER, PR, and HER2. I've discussed this with you yesterday, HER2 in particular, but estrogen receptor and progesterone receptors are also hormonal receptors which are expressed in ductal cells, and their overexpression can confer advantage to the proliferation of the ductal cells and leading to ductal carcinoma. PR, progesterone receptor, if it is overexpressed, then it would be a good prognosis to such kind of a patient. So in this scenario, we did a cytology, and this is why you see very few cells running from now the where you see the cytology hematoxylinucin to come now to the markers here. The first one we have the PR, the second one we have ER, B is the PR, C is the ER, and then the D, the HER2. I've got a slide here which is showing you a clear picture of now HER2 analysis in the tissue biopsy, not now coming from the cytology. Nonetheless, one thing I need to clarify for you here is PR and ER is detectable in the nucleus of the cell. HER2 is detectable in the cytoplasm of the cell and also the cell membrane. So you see each cell here is separated from one another because of the protein deposit of the HER2 protein. Later, When I'll be taking you now through in situ hybridization, then we'll be looking at this gene at the level of the nucleus being amplified there. But today, we are going to look at the protein itself expressed on the cytoplasm and the cell membrane. So typically, these things have got clinical relevance because a patient who is HER2 positive, 2 plus, is a potential patient who can benefit from monoclonal antibody therapy, which I taught you yesterday. A patient who is progesterone receptor positive and ER positive, this patient is potentially going to benefit from the anti-ER targeted therapy using tamoxifen. So you need to learn all this and keenly ensure that whichever laboratory, whichever pathology department you'll be working in future, this must be done correctly. Because if you do it wrongly and a patient is treated with anti-ER, the tamoxifen, yet the test was not done correctly and it was negative, you are injuring the system of that patient. If the patient was diagnosed as ER negative, yet this patient was to be ER positive, then you are doing disadvantage to this patient who could have benefited from anti-hormonal therapy. So other examples in the utility of immunohistochemistry, you have got soft tissue tumors profiles. You can make diagnosis of immunohistochemistry in soft tissue tumors, although not much applicable. Most of the time, you have the CD99 right now, which is available to make soft tissue tumor diagnosis. But the WHO clarifies that there's not much clinical application in the diagnosis of soft tissue tumors, and particularly the sarcomas using the immunohistochemistry. However, diagnosis of the carcinomas, and yesterday we were talking about, say, gastric carcinoma, nasopharyngeal carcinoma, or gastrointestinal tumors, which are associated with the infectious agent, say H. pylori or Epstein-Barr virus infection in the gastric, you have got a lot of the clinical utility of the immunohistochemistry in making the correct confirmation of the diagnosis of these carcinomas. There are also the lung carcinoma. We have got also pituitary carcinomas. We have also the nasopharyngeal carcinomas. You have got the breast carcinomas. All these need your keen application of the immunophenotyping or immunohistochemistry to make sure you make correct diagnosis. Note, do I need to repeat the hormonal receptors in breast cancer and also the HER2, which if you diagnose correctly, you are really saving the life of a patient who is lying there in the ward. And I think up to that end is what I want to end with you, because you are not in the technical field, so I don't want to go into the critical steps of the immunohistochemistry. This you'll be learning them when you'll be interacting with the technical staff on the bench. But so far, we've looked at the immunohistochemistry, its principle, and the clinical application, unless any student has a question. What we have covered, then you can pose the question. You have got a case in histology, you look at the methoxylin eosin, how it goes, and then power you can increase up to higher power. Then you start looking at, are you able to detect these pathogens which are associated with this tumor or the markers which are expressed on the surface of that tumor when you're using immunohistochemistry. So when we'll be coming to in situ hybridization, not only I've mentioned to you the in situ hybridization, I'll go into detail into it. I don't want to go to detail today because today we are covering immunohistochemistry. But in situ hybridization can as well help you detect the gene itself, like we see in HR2, as well as the pathogen itself, like here being the detection of Epstein-Barr virus encoded RNA in tumors, which in cells which are affected with this virus. So instance of, I've also mentioned to you the HHV8 Kaposi sarcoma associated herpes virus, its detection in the tumors. But for the case of the HHV8, you use just a monoclonal antibody, which is detecting a protein which is expressed by that tumor, LANA, latency associated nuclear antigen. I don't know whether I have any here. Here. So this is for the detection of the HHV8 using the immunohistochemistry. Like I mentioned to you, you can have primary effusion lymphoma, which are associated with the HHV8 or Kaposi sarcoma associated herpes virus. Now, if a biopsy or pleural effusion is extracted and you prepare those cells well, you can as well use immunohistochemistry to detect the pathogen. So this is a third clinical application of immunohistochemistry, that you can as well detect also the pathogens. We saw the utility in the breast cancer markers. We saw the utility in the immunophenotyping, like for the case of carcinomas and lymphomas. But here I stressed your point also that immunohistochemistry as well can be used in the detection of pathogens which are associated with these tumors. So from there, I think we end it there. Next time when we'll be covering, we go now to the other molecular techniques, which include the PCR, in situ hybridization, and flow cytometry. Any question?
Doc, I had a question.
Yes.
When a biopsy is done and it's requested for histology, is the immunohistochemistry done routinely, or what is it they exactly do? Like in detection from any biopsy, if it's being suspected for malignancy, is immunohistochemistry done routinely?
In advanced laboratories, and especially for the immunocytology, internet connection is unstable. Well, most of the time, to put it as a package so that you don't ask the patient later, like you can't pay for immunohistochemistry, it is configured inside there. So you as the pathologist is the one to look at what raft of tests do I need to do to make the correct diagnosis of this tumor. You get me? So it should ordinarily be part of the package of the histology analysis. But in our setup, it is not running like that because of limitation of the resources. So it's also therefore compromising the quality of diagnosis.
Oh, okay. So in Kenyatta, they don't usually do the immunohistochemistry.
The KNH, they do, but they only do for the case of breast cancer receptors because the Roche pharmaceutical company sponsored that.
Oh.
And the other markers, I see we are still struggling to sustain the purchase or supply of the lymphoma and also carcinoma markers, monoclonal antibodies.
Okay.
So like I mentioned to you, it confers disadvantage to the quality of the diagnosis to the patient.
Yes, yes.
Because sometimes when you have got metastatic carcinoma from one site to another, when you use these markers, which I've mentioned to you, you learn with time, you can use the markers which are expressed by the tumor cells to locate the origin of the tumor. And so it is not necessarily at the site of metastasis, so you can locate the origin of the tumor using the immunohistochemistry markers. But if you don't do that, definitely you'll be just querying, this is squamous cell carcinoma, and somebody may ask you from where. You can be diagnosing metastatic breast cancer is from breast, but you can be talking about metastatic carcinoma. Then you leave the diagnosis like that. I mean, a qualified or an experienced pathologist would ask you metastatic from where, so that that primary organ can be targeted either for radiation therapy or other targeted therapy.
Right. Thank you, I've understood.
Well, if you don't have any more questions, we can break and then see how we can continue in the next lecture, which would be